What else do we know? Ooh, 2 nuclei. Very good. So we're just starting the recording. As students are coming in, we're looking here at the board at characteristics of mitosis. Okay. Ooh, very good. PMAT. Brianna, you're on fire today. What else do we know about mitosis? Got some awesome characteristics for sure. That is so good. I'm so glad you said that. Brianna said it's fresh from yesterday's lesson, which I'm going to say it's probably not most of the people that are in this room. But I hate being sick, okay? I know, I hate being sick. I was sick. Both of the girls had fevers. We were not feeling well. Yeah, I don't know what it is. We have a little bit of congestion, but we're bouncing back pretty quickly. But I was not able to be here yesterday. So what I did is I gave you a really quick Nearpod, a Nearpod that had like less than 7 minutes, right? A 7-minute video and a review game. There were 18 people that actually watched the video out of 150. That's right, Riley. 18 out of 140 watched our lesson for yesterday, and it was only an 8-minute lesson. All right, instead of a 45-minute class, there were only 18 people that watched a 8-minute video. It's in the Nearpod. It was the Nearpod. It was the Nearpod, and then the word wall game. You want to know how many people did the word wall game? Less than 18. 3. 3 people did my game. So the review game, I put it in the announcements and I put it in your plan for yesterday. Let me find it. What's in it to go? Come back. I can get you the link for it. I'm not trying to be that person. It just made me super sad because I'm here most days. So I just was a little bit sad that on a day that I needed you all to do a short lesson without me, for some reason. So here is the word wall game. Right. Again, I'm not hating on anybody. It just made me in general very sad. And I was giving you those lessons. I actually came up with those lessons yesterday while I was sick, while I was taking my day off, so that you guys wouldn't get behind. Okay. And now we're gonna have to go back and review some of that for those of you that didn't do the work. So I am going to spend a little bit of time on it today, but those of you that did it, I can tell you did it because you are fresh with some of this stuff. Okay. And again, I'm not trying to like be that person and put a bunch of guilt on you. But that's how it is with eSchool. We don't have substitute teachers that come in. And honestly, substitute teachers can be, right? So sometimes you'll have a good substitute and sometimes you won't. Sometimes it'll be meaningful, sometimes it won't. So I tried to make it a short, meaningful lesson for you yesterday. So I hope that in the future, if I have to make if I am sick and I have to make a lesson for you instead of be available, even for like testing. Ooh, that sounds exciting, JB. All right, that you'll come in and do your work because I really do check. All right. And that's not to put guilt. It's sometimes we just have to take responsibility for our learning. And that is my talk for today. I'll be done with that.
He needs help.
He needs help, Dad. You gotta pick him up.
You can't lift him up by his armpits or he yelps.
I know he has a hard time.
We got somebody with their mic on. Everybody okay? Want to just have you check your mic?
Come on, Hunter.
You got it. You did it.
Let's see. Got a microphone on, which is totally fine if you want it on. If you don't want it on. And Caden's got a sweet hat on today. JB's got a new desk. It's gonna be a good day in biology. That sounds cool. Carbon fiber and steel. That's awesome. Jana got a puppy. Are you able to put it on camera, Jana? Dobermans have such a cute face. Oh, a kitten soon, Diamond. That'll be fun. I know. I'm looking for Jana. Are you going to show us that cute little pup? Oh. Babies bring such joy, Diamond. A little baby kitten might be just the thing you need. Awesome. Well, for those of you who just are joining, we worked earlier on a Freyer model that we looked at all of the examples of, or non-examples, characteristics. We had very few that participated, and so that tells me we may not know that much about mitosis. We're going to briefly hit on it today, but today is also our meiosis day. So if you are here and you did not do our lesson from yesterday, I'm going to encourage you after class or after your scheduled classes today, put time in your schedule to go back and do yesterday's lesson. It's a good video on what mitosis is, and there's a word wall that will help you review. All right, so do that if you need extra help on mitosis. We are going to finish up Unit 3 this week. All right, we're here today and tomorrow, and then I have help sessions. Awesome. Here we go. Anybody want to read our goals or objectives for today? What are we going to do today? What are we going to be able to walk out of here flexing our muscles saying we can do?
Today?
Dutch. I'll be Arthur Moore. What is mitosis? What are the phases of mitosis? What is meiosis? Are you able to write the difference between mitosis and meiosis? Okay. Why does meiosis important to reproduction, Dutch?
We have got Caden with his strong accent going well this morning. Thanks for reading, Caden. So yes, we are going to be able to tell what mitosis is, what meiosis is, and we're going to be able to kind of make some differentiations. If you're not able to do this by the end of the day, then we're going to make some goals on some steps you can take to get there. How do we feel about mitosis and meiosis? Give me a quick poll right there. Mitosis, and I like to say meiosis. All right, 15 of you answered. Some of us are feeling good, some of us are feeling about it. So let's go through today and really use our time together wisely. right? This picture is here to show you one thing, all right? What is one thing, what's in your day that you do not spend a lot of time doing? You do it every day, but you do not spend a lot of time doing it. I know, I want to see that puppy too, JB. What's something you do every day? Oh, there he is. There's the puppy, JB. Oh, Jaina, he is adorable. Scroll to Jaina's camera. I presented her. Can you see her now? She should be up presenting on your screen. Let me see. I can see her. I'm not sure why you can't. I put her up as I was muted too. Thank you. What is something you guys do in your day that you don't spend a ton of time doing? You do it every day, but you don't spend a ton of time doing it. Eating. Check your email every morning. Eating cheese. School. Awesome. So what happens in the life or a day of a cell is that there is a very short period of time in which the cell actually goes through reproduction. Okay? If you look here at the chart, it spends a lot of time growing. It spends a lot of time copying its DNA, all right, making a copy of its DNA. And then it spends a little bit of time getting ready. But look at this, 9 hours of growth, 10 hours to reproduce the DNA, 2 hours to prep, and it only takes 1 hour in general to actually go through cell division. Kind of like, I don't know, brushing my teeth. I brush my teeth in the morning, I brush my teeth at night. Something I do every day, but it doesn't take up a ton of my time. So I want you to kind of think about this. Although we spend a lot of time in science talking about it because it's a very important process, in the life of a cell, it's an actually very short period of time. So you tell me, does it take more time to get ready to divide or to actually divide? Say that again. Does it take more time for the cell to divide or getting ready? Yes, getting ready. You're absolutely right. It takes way more time to get ready. All right, Gina, I'm trying to make you not presenting now. But I'm not doing a good job of putting you back. I'm trying to make you go back down. You might just be presenting a little longer. Awesome. All right. So we spend most of our time getting ready. Let's now look here at the cell theory, right? The cell theory we learned earlier in the unit, and it's that all cells come from where? Go ahead, everyone type that in the chat. All cells come from other cells. Perfect. All right, so how do we do this? Well, we are going to go through interphase, into mitosis, and then into cytokinesis. That's important because interphase is often grouped into mitosis, and it is actually not a phase of mitosis, it's the prep phase, all right? Interphase is the stage before mitosis. What happens in interphase? JB, would you read what happens in interphase?
Quite gladly. This stage is before mitosis. The cell has to grow and produce all sorts of stuff that happens for basic survival, and it has to make a copy of everything inside of its nucleus.
Perfect. You said it perfectly, JB. All right, before a cell can divide, it needs to gather all the stuff. Just like when I go to cook something, I go get my measuring cups, the bowl, the recipe card, the ingredients, all the stuff, right? It's the same thing for mitosis. In order to go through mitosis, we got to go get all the stuff. Right? And that is going to happen in interphase. The most important thing that in that entire process may actually be copying the DNA. Can you make another cell without a copy of the DNA? No. And if you did, it would not survive very long. So funny. Awesome. So good. That is interphase. It's the phase prior to mitosis. So the first stage of mitosis would be what? Anyone know? Yes, Brianna reminded us this morning so well, PMAT. All right, so if you need a acronym to help you remember the stages of mitosis, it's going to be PMAT. Everybody type PMAT for me. Capital X P M A T. PMAT. All 57 of you should be blowing up my chat saying, PMAT, I'm here, Mrs. Rittenhouse. There you all are. Jerry. That made me laugh. Awesome. All right, so the very first stage of mitosis would be with the P for prophase. Prophase. All right, I love to do drawings. If you are a person that is good at art or even just likes to get artsy, this is a great lesson to do it with, okay? What happens during prophase? Can someone read what happens in prophase? Any volunteers?
Sure. Okay, nucleus prepares for division by beginning to break apart. This allows chromosomes to become free floating in the cytoplasm. All chromosomes copies are held together at the centrometer to create an X formation.
Thank you, Kaden. High five. So yes, what happens? I'm trying to think of a good example of this. Kind of like those. Have you ever had those chocolate eggs? Or like, anybody have those hot cocoa bombs? Last winter, like those things like were super popular. Like you stick it was called a hot cocoa bomb, and you stick it in warm milk and it melted. And then I had some that had like marshmallows and sprinkles that would come out. Anybody else have a good one? I had a peppermint one that had like candy cane. Basically, why are we talking about hot chocolate bombs? Because it's kind of like that in prophase. What happens is that nuclear membrane around the nucleus of the cell, it like dissolves. All right, so that nuclear membrane dissolves, and what you see are these thick chromosomes. So we talked about before, like think about yarn. All right, our DNA, let's make sure chat is not getting too off base just because I don't want you to miss this. It's so important. All right, so if you have yarn, just like in a wad in your living room on the floor, a whole, like, it's not a spool, I always call it a spool, a whole ball, is it ball, of yarn is unspooled on your living room floor. That's what it's usually like in the nucleus. All right. When your cell goes to divide during interphase, it actually loops it into a ball. It makes them into these little, like, chunks of, they look like little French fries of DNA. All right, that DNA is wound into short. You're fine. I don't know what's going on in chat. It's okay. Hope everyone's okay. We're all chatty today, and that's fine. I'm just trying to bring everybody back in. Okay? So in prophase, you have the DNA French fries, right? The DNA that's all wound up, and that membrane of the nucleus dissolves so that those chromosomes have room to move throughout the cell. The next phase is metaphase. All right, now we're going to organize our French fries. Who can read for metaphase for me? Anyone want to read?
I will.
Thank you.
The chromosome lineup in the center of the cell. The spindle fibers attach the two chromosomes at the centromere, where they join. The
Perfect.
engineer Gaiman.
Yes, so metaphase is going to be where you see in a cell that they are lined up down the middle. All right, so the copied DNA and the original DNA find their, what they call, sister chromatid. So it's like, oh, hey, you code for eye color. I code for eye color too. Let's line up together. Does that make sense? Kind of like your chromosomes play matchup. Your new DNA that was copied and the old DNA that was in the cell find their sister chromatid. They find their matching pair. And there's actually something called a centromere. And a centromere reminds me of like a stapler. Once it finds its match, they kind of staple together and they look like an X. All right. And then those pairs are lined up down the middle of the cell. So they find their match, you staple them together, and they line up down the middle of the cell. All right. Does that make sense to everybody? Give me a green check, red X. In metaphase, they line up down the middle. In metaphase, they line up down the middle. I know, Jane is still presenting because I don't know how to make her not present. Sorry, Jana. I wanted you. Oh, we switched spots. Is that helpful? Awesome. So we're feeling good. You can tell Ren's feeling better today, aren't you, Rennie? Awesome. She's watching 101 Dalmatians. So an anaphase. Anaphase, we're going to take our matches and something is going to happen. We don't need two chromosomes. We take the two matching chromosomes. I don't need two chromosomes for eye color. All right, but the other cell is not going to make another functioning cell without that set of DNA. So what happens in anaphase? Can someone read for me? What happens in anaphase? We're going to take those matches and do something with them. Who can read for me? Please, please, please.
Yeah. I'm watching you, baby. I'm watching you. Anaphyse. Spindle fibers begin to separate. How can I look down on all of you? Anaphase spindle fibers begin to separate chromosomes. I'm judging you. One complete set is smear on my computer is pulled to one side, and the other complete side set that I can't see like this. Complete set is pulled to the other side of the cell. Separation.
Yes.
This ensures each cell has the exact same genetic material. Identicality.
Perfect. Way to go, Kaden. Thank you. So yeah, what happens now is we take those matches that we just put together, and that helped us organize what we need in each cell. We keep hearing about these spindle fibers. I like to think of these spindle fibers kind of like an anchor. Okay, so these anchors form on both ends of the cell. There are two anchors. And the chain from each anchor is going to each one of those chromosome sets. Does that make sense? So those spindle fibers are kind of like the chains to the anchor. And so an anaphase, I kind of like to think of it as a girl with pigtails, right? Because they're pulling the chromosomes of one of each kind. So eye color and eye color are getting pulled to the opposite side. Height and height are getting pulled to the opposite side. Let's say hair color. All right. All of your different DNA traits are getting pulled from their match, right? They found their identical match. Well, we need one in each cell. So those spindle fibers are pulling those chromosomes apart so that we can end up this very last phase of division called telophase. All right. Telophase is the last phase of mitosis, and then we have cytokinesis. All right. Telophase is what? One last reader for the day? Yes, telophase equals 2. I like that, Brianna. Ooh, anaphase is away. AA, they pull away. And anaphase, they pull away. And telophase, they have 2 cells. Good. So what happens is the cytoplasm actually during cytokinesis, so once the DNA gets to both sides of the cell, they've completely pulled away from each other. Okay? Then our cell actually cytokinesis. The best example I have for this is like a balloon, right? Whether it be a water balloon or a balloon you blow up, if you twist it, right, it makes that pinch off into 2. Cytokinesis literally is a pinching of the cytoplasm until one cell becomes 2. Right? In this picture, you can see that the DNA has been pulled apart, and you can see right here this new cell wall is coming into place. All right. Good. So those are the phases of mitosis. PMAT, prophase. All right. That's where your DNA is condensing into your little French fries of DNA. Metaphase. Yes, those chromosomes come to the middle and meet up with their identical pair. All right, and anaphase, they pull away from their pair to each side. And then telophase, they become 2 cells. And the cytoplasm finishes it and cytokinesis by pinching out. Ooh, a bread tie, JB. I like that. Like a bread tie. Super clever. So telophase is the last phase of mitosis. Cytokinesis is a separate process. Cytokinesis is specifically the pinching of the cytoplasm. I know, is Alex here today? I miss Alex. Good. Awesome. So that is mitosis. I'm going to come back to this in 1 minute, okay? And we're going to identify some of the cells in this picture. Like, where's Waldo? Do you guys know? Where's Waldo? Yes. All right, we're going to play Where's Waldo with ourselves. All right, here's where we're running out of time because we didn't all do our lesson yesterday. Because no one loves me and didn't do what I told them to do. No, and it's not everybody. Sorry. Get emotional there. Awesome. So, what we are going to kind of miss out on, and we might need to spend some more time today in, is meiosis. Meiosis. All right. It's different than mitosis. Mitosis makes those diploid cells that we talked about on last Tuesday, Wednesday, right? Our lung cells, our brain cells, our stomach cells, all the cells of our body, minus our sex cells, our reproductive cells, right? Those cells are made through meiosis, like me. I was made by meiosis, two cells that were made from meiosis. Mitosis makes diploid cells, meiosis makes haploid cells, all right? So mitosis starts with a diploid cell and ends with two diploid cells. Meiosis does mitosis 2 times through. All right, it goes through interphase, and then it goes prophase, metaphase, anaphase, telophase. All right, and then those 2 cells that it produces, all right, these are just like what mitosis ends with. These 2 cells go through division again, but they don't do interphase, right? Because interphase is not part of it. Prophase, metaphase, anaphase, telophase, so that we get half the number of chromosomes in these cells. Okay? So that's what we're going to need to spend time on today, is going back and looking at the difference between mitosis and meiosis. I will spend a tad of time tomorrow on it, but I did these lessons so that we wouldn't get behind. All right, so we're going to try to finish this week strong so that by Wednesday, tomorrow morning, we feel good about what mitosis is, what meiosis is, and be able to compare the two. Okay? So, tomorrow we will work on these. Right? What is the starting and what do you end with? I want you to draw over here what you'll end with. No, I'm not telling you you have to read. I don't learn super well by reading. Reading is probably a good thing for most of us to try to do, but I did post a video yesterday. Watch that video. I will post another video today. Okay. Awesome. So as we end class today, what I want to do is play some Where's Waldo with these cells. When I move my face to presenting, does it move it for you? No? Am I still in the left-hand corner covering? Okay. Oh, you could move it on your own. Move it on your own so I'm not covering your little cells over here. Okay? All right. So I want you to find. Actually, somebody tell us a phase to find. Pop on your mic. Tell us a phase we want to identify. One cell of mitosis. One phase of mitosis. Cytokinesis. Okay, somebody help me find one set. First we'll find cytokinesis. Next we'll find metaphase, Brianna. Ooh, yes. Whoever this person is correct. Look, you see the pinching. There should be some more. Find me some more. There should be lots of pointers on here. Let me see lots of pointers showing me a cytokinesis cell. Good. Cytokinesis should be one of the more identifiable because you'll see the whole DNA in both sides and you'll see it pinching off and you'll see from telophase that cell membrane kind of repairing itself down the middle. Good. All right. Brianna says, give me a metaphase. Let's find a metaphase cell. Yes. I don't know why metaphase is like my favorite. I just feel like I like that it's so identifiable. I like clarity. All right, let's do one more. Let's do anaphase. Anaphase. They're pulling away from each other. All these pointers are making me so happy. Yes. Yes. They're pulling away. Super pumped. Awesome. All right. So I want you to think on this today. All right. Some of these objectives we accomplished. Some of them we did not. Take a peek at these. What is mitosis? What is meiosis? What are the phases? Point to the ones that you feel like you're the best at right now. I feel like a lot of you are good with mitosis and the phases of mitosis. I know you have. I see you, Peyton. All right, so knowing what we have just identified as our strength, we now see what our weakness is. All right, meiosis and knowing the difference. So what are you going to do today? What are you going to do today to move a step forward? All right, because I'm here to help you for class, but it's going to take a little bit of accountability on your own part. Are you going to read? Caden is not going to read. All right, maybe he's going to complete some assignments. Maybe you'll do that word wall game. I'm going to put that in chat again. I'll put it on the board. Maybe do that. Maybe you will watch the Amoeba Sisters video I'm going to put up of meiosis. Okay, I'm going to put a meiosis video up today in the classrooms on the announcement page. All right, what are you going to do? I want to see lots of pointers. Maybe you're going to make flashcards and you're going to do drawings of the phases. All right, maybe you're going to do a flow chart of mitosis and of meiosis. Or maybe you're going to go back and watch today's lesson. I love it. Taking notes is perfect. Awesome. So we are going to start using these slides more often to help us figure out what we know, what we don't know, what we're going to do about it, right? Awesome. Well, listen, I appreciate you all for being here so much. We're going to do some covering of meiosis tomorrow. We're going to look at the differences tomorrow. Do your part. I'll do my part. I'm going to come up with some videos to help us and have a fantastic day. Thank you guys for being here and participating. Bye, guys. You made my day. Thank you. Enjoy that sweet puppy, Jana. In your new desk, JB. Tell your pups we said hello, Kaden.